What we are trying to do in the lab is to use model organism, and especially the mouse, to understand whole organism physiology. And the rationale for that is that physiology has an old concept, whole organism, how organs function within the body, has been given up in the late 50s for molecular physiology. And there is a need now and a possibility with mouse genetics to knock out one gene at a time in a cell type of interest and in a reversible manner that allows to revisit whole organism physiology and to see what is the impact on pathology, whether it's linked to aging, as it is the topic today, or not linked to aging. So obviously you cannot do that in a single lab in looking at all organs, and we study in my lab one single organ, which is bone. And using genetics to study bone physiology for us means to ask the question, does bone has any other function besides making bone? Which also underlying question is, why did evolution invent bone? So I know as a fact, because I am the author of the paper, that you can ask this question for any organ, fat, liver, kidney, do they do anything else than what they are known for? But you cannot ask it for bone. And the reason is not scientific or medical, it is much stronger, it's irrational, and it is cultural. And the reason is that if you want to represent death without using the word death, in any country of the world, that's what you will show. And that you know that people are speaking of death. And if you want to present the danger of death without using the word danger of death, that's what you will show. So the association, cultural association between bone and death, or bone and inert tissue is so strong that it is something that we have to overcome, because we know that from an evolutionary point of view, obviously bone was not invented to become a cultural symbol. So, in fact, bone has one particular cell characteristic that is fundamental, and it is the only organ or tissue in the body that contains a cell type whose only function is to destroy the host tissue. It's as if bone has invented autoimmunity, but it's a physiological process. In fact, it's half of a physiological process. The physiological process is called bone modeling during childhood, and the results of bone modeling is not inconsequential. And in adult, it is here to repair micro and macro damage. In other words, until the beginning of the 20th century, bone remodeling was the only orthopedic surgeon around. Regardless of the function of bone modeling and remodeling, committing suicide every day in hundreds of locations like bone does is quite expensive energetically. And it's even more expensive if after that you decide to be reborn. Whatever is the cost of energetic cost of bone modeling, remodeling, it is obviously proportional to the area covered by the organ. And bone covers more surface than the retina, the spleen, or the lung. So the cost of bone remodeling is quite important. This concept is in fact verified by the best experimentalist that we know, which is medicine, because any child who will not eat, whether he is or she is anorectic or does not have access to food, the first symptom will be an arrest of growth. If there is no energy intake, there is no bone modeling. And in adults, anorexia nervosa causes osteoporosis. And we also know that gonadal failure in both sexes causes bone loss. So to use mouse genetics to ask the question, does bone have any other function besides making bone, we came up with this hypothesis. There should be a coordinated control, endocrine in nature because bone are not next to the gonads, of bone growth, energy metabolism, and reproduction. I'm not going to tell you or to give you the history of the lab, but the point is that if this hypothesis is true, bone cannot be only a sponge receiving endocrine input. It has to be an endocrine organ regulating energy metabolism and reproduction. And I think here there is a need to introduce definition. How do you define an hormone in the era of mouse genetics? And its function should be detectable in both loss and gain of function model. The success of insulin is that if you give it to people on top of it, it has an effect. The function should be detected on a normal diet because high-fat diet was invented by McDonald in 1952, but evolution started before that. It should bind to an identifiable receptor, and preferably it should function in primates. So with these criteria in mind, in fact, we found an hormone made by bone, which is called osteocalcin. It's the most abundant protein made by bone besides collagen. It's in fact one of the most abundant proteins in the body, but most of it is in bone in an inert form. And for other reasons, we decided to knock out osteocalcin at the beginning of the 90s, and we obtained a mouse with no phenotype. So the first reaction is to say, well, it's bad luck. Maybe you should go to another gene. But in fact, it's impossible. It's impossible that evolution went as far as inventing a gene that is so highly expressed in a single cell just for the fun of it. And osteocalcin has all structural features of a hormone. And to study osteocalcin, and this is where we were lucky, we had access to a loss of function model, the osteocalcin knockout, and a gain of function model. Doesn't matter what is the gene, they just have more osteocalcin. And using these two models, we could show, before I go in what osteocalcin does, there is another aspect important of osteocalcin which probably justifies why I am here, is that circulating level of osteocalcin, the active form in the open square, is virtually gone at 9 months in mice, in females, and same thing in male. And this is not a mouse story, it's the same thing in women. Women has low osteocalcin at 20-year-olds and at 78 years old. In other words, osteocalcin has imprinted in it the evolutionary clock. Women 1 million years ago did not need to live to 35-year-old to give babies, to give birth. And most of the most important cause of death of women at that time was childbirth. And men were not really in better shape. So this is the evolutionary clock is imprinted of osteocalcin, but since an hormone is important only if it has a gain of function phenotype, it also be important for an aging purpose if we could rescue the function I'm going to describe. And so osteocalcin, we could show, is upstream of insulin and glucose metabolism. We identified the receptor, we knocked out the receptor. It's upstream of male fertility, and we could show that it is true in mice and in humans that have a mutation in this receptor. And so this fulfilled the hypothesis, and there is more molecular work to do. But the question, it was intuitively obvious that osteocalcin was not invented by evolution to fulfill the prediction of a scientist, even if the scientist is French. So it must have other functions that were not thought about or anticipated. And one of them is that circulating level of osteocalcin triple or quadruple during exercise. It's endurance exercise. It's true in mice, and it's true in humans. At the same time, circulating levels of insulin decrease. And it's important because you could think that there are, in fact, two physiological settings. One is resting, which is the one we do every day, and one is exercising, which is what our ancestors were doing 20 hours a day, more or less. And adaptation to exercise is a conserved function that relies on the ability of muscle to break down glucose and fatty acid, sugar and butter. Just by saying that, it rules out that insulin is involved in the control of exercise because insulin is anabolic. And it favors the uptake of glucose, but the storage of glucose, which suggests that there are circulating molecules. They can be cytokines, myokines, hormone, but circulating molecules that act on muscles to increase muscle function so that we can run longer and faster. And since this work has been published, what we showed is that in mice lacking the osteocalcin receptor in myofibers only, there is a 30% decrease in the ability to run for these mice. So 30% doesn't seem like a lot, and in fact it's not a lot, except if you are a mouse and live in Central Park, and it's probably going to be your last day. And it's true also in compound heterozygote mice lacking one allele of osteocalcin and one allele of the receptor, indicating that this is a function of osteocalcin, and indicating also that there is only one ligand for this receptor, it's osteocalcin. Because an hormone has to be necessary and sufficient, and because osteocalcin is gone in aging, we ask what happens if you give back osteocalcin, but now to wild-type mice. So wild-type mice put on a treadmill runs approximately, at 3 months old, run 1,000 meters. If you give them osteocalcin just before the experiment, they run 25% more, which is not bad, but that's what my postdoc, and I don't know why she called it that, she is Spanish, she called it the doping experiment. Then we did it in 12-month-old mice. They run only 600 meters. And now they run 1,000 meters. In other words, they are 3 months old in terms of muscle function. We did it in 15-month-old mice, and it's the same thing. We did it in 18-month-old mice. It's the same thing, then we stopped. In other words, osteocalcin can reinstate function in muscle of a 3-month-old mice up to 18 months in age of the mice. And therefore, it is necessary and sufficient, at least in muscle. So that's the first function that we identify, but then we look at the mice with more details, and we look at more function, and what we observe is that osteocalcin is necessary for memory because it acts through another receptor in the hippocampus. And we all said this morning, we heard it's necessary, it has a function. Those are neutral words for us, scientists. I think it's better to represent exactly what the absence of osteocalcin, which is synonymous of the absence of bone at the level of endocrinology, has on memory. So it was working a minute ago. A wild-type mouse has to find the platform because mice hate water. They are blind and the water is milky. So it's only based on memory. You can see that the mouse that has no osteocalcin, in endocrine terms has no bone, it will not go to medical school this year or next year. In fact, it will never find the platform. What you see is the influence on the skeleton, on the brain, on memory, okay? So, well, I'm not going to let it go because we could be here tomorrow. So, again, how to define an hormone is a gain of function on a normal diet. And so we went back to this experiment that Darren spoke about, which is the parabiosis between an old and a young mouse. In fact, it's not a parabiosis in that case. You inject plasma of young mouse in an old mouse, they become smarter. It looks magic. And there is, at that time when this paper was published, there was no molecular basis for that. So we reproduced this experiment, but this time using old and young mice, wild type or osteocalcin knockout. So if you inject the plasma of young mice in old mice and use an elevated plus-minus test, but we use also the novel object recognition, the test does not matter. The result is mice become smarter. The mice here are all wild type. If you now inject in a wild type old mouse the serum of a young osteocalcin mouse, it will not go in medical school or anywhere. It's stupid. If you take this serum of a young osteocalcin null mouse and inject it with osteocalcin, now the mouse can go to medical school. And it's important, this experiment, because it shows it's not developmental. It's all osteocalcin. And again, it shows that for memory, osteocalcin is not only, or bone, is not only necessary, it is sufficient to restore it. This experiment was done at different age, and the mice were 16 months old versus 3 months old. Okay, so now we have another problem. Osteocalcin is upstream of insulin, it's upstream of testosterone, it favors exercise, it makes mice smarter. And the question is, what is common between these functions? How an hormone can have so apparently disparate function? What is the logic behind it? And which is another way to ask why bone when you want to study if this organ has other function. And so we looked at that in great pain because it's not obvious what is common between memory and exercise or between testosterone and insulin. However, memory was not invented to go to medical school. It was invented to remember where was the predator an hour ago or the food yesterday. And if you live in the Amazonian forest today, and if you don't remember these two parameters, it's going to be a very tough day. But even if you have an outstanding memory, if you cannot run, I think you are dead. So, and when you run, something happens that is not explained. Circulating testosterone goes up. So, under this light, and I agree it looks like an impressionistic hypothesis, but it can be tested. Oops, what did I do? Most functions promoted by osteocalcin are needed to overcome danger, acute danger. And you cannot say that of any hormone. If a lion comes here, whether I have prolactin or not will not change anything. Same thing for PTH or erythropoietin. If this hypothesis is true, then osteocalcin should regulate other functions needed for escaping danger. And the quintessential function to escape danger is the acute stress response. So the acute stress, and you don't need estrogen, prolactin, LH, FSH for the acute stress response. So the acute stress response is an evolutionarily conserved physiological process that maintains or restores homeostasis in front of an acute danger. It manifests itself by a myriad of manifestations, but you cannot study all of them in mice. So the one we focused on are vasoconstriction, heart rate, respiration, and energy expenditure, and they all go up. And if bone, oops, sorry. Oh no, something happened. Anyway, there is a paradox in the acute stress response. I don't know if it's a paradox or if it's me who did not study medicine the right way, but what we are told in medical school and elsewhere is that the sympathetic tone and glucocorticoid hormones are mediators of the acute stress response. At least that's what I remember. And there is a major problem with that. If a lion comes here, it will trigger an acute stress response within seconds, minutes. Whereas glucocorticoids are steroid hormones, and they need hours to act. So there is a problem. So if there is an endocrine mediation of the acute stress response, it cannot be anything else but the peptide hormone. And of course, if bone is a survival tool, it has to be implicated in the acute stress response. And so in closing, I will show you a work that we just published yesterday about bone and the acute stress response. In fact, bone, adrenal gland, and the acute stress response. So there are two criteria that define a stress hormone. One is that it should go up during stress, like steroids. But the second one is that it should enhance physiological process recruited during an acute stress response. In other words, if you don't have the hormone, you don't have the stress response. So we tested many stressors, but I'm going to show only one for the sake of time and also for the clarity of the experiment, because we know these stressors act in the brain. It's fox urine, a compound of the fox urine. And if you expose mice to fox urine, within 2 minutes, you have a 4-fold increase in circulating osteocalcin in blood, 4-fold. And it precedes the increase of glucocorticoid, and it lasts approximately, at least 3 hours. We stopped the experiment because after that, it is confounded by the circadian rhythm of osteocalcin. It's also true in rats, and if you ask people to give a talk without warning, there is an increase in circulating osteocalcin. We did this experiment. Not me, but others. If you destroy the neuron on which fox urine work in the basolateral amygdala of the brain, and then stress the mice, they are zen. There is no stress response. That was known, and there is no increase of osteocalcin. So the question is it needed for the stress response? Is it a stress hormone, or is it just like glucocorticoid, a marker? So we looked at that in osteocalcin knockout mice. We stressed the osteocalcin knockout mice, and it's a little bit like memory. You can scare them to death. They don't care. There is no stress response measured by energy expenditure, heart rate, respiration in the absence of bone, of osteocalcin, which is just a vector of bone influence on other organs. It's a peripheral function of osteocalcin because if we do the same experiment in mice that lack the receptor of osteocalcin in peripheral tissues, they don't have a stress response by any means. So the ultimate mediator of the stress response is the sympathetic tone, and the sympathetic tone is in equilibrium in you and I with the parasympathetic nervous system. This is the flight and fight tone. This is the couch potato rest and digest tone. So we tested by electrophysiology and by other means, but I'm not going to show all the experiment, where osteocalcin acts, because osteocalcin cannot by itself mediate the stress response. And so if we inject osteocalcin in mice, we have no effect on electrophysiology on the sympathetic nervous system. On the other hand, we diminish the parasympathetic nervous system. In other words, we relieve the brake on the fight and flight response, and now there can be a stress response. And accordingly, it's also true in humans. If you look at wild type and osteocalcin mice, if you stress them with fox urine, in wild type mice, the parasympathetic tone goes down, and then the fight or flight response can go, the brake is off. In the osteocalcin mice, it's exactly the opposite, and they are zen. Okay, so what about the adrenal glands in this picture? okay? So to address this question, we used wild-type mice in which we remove adrenal glands. So we remove glucocorticoid, mineralocorticoid, and epinephrine. And we stressed the mice. We did the same in rats, and the experiment has been done in humans, not by us. So if you take adrenalectomized mice and stress them, these mice have a normal stress response by any criteria. Heart rate, blood pressure, energy expenditure, and respiration, I think, yes. You do not need the adrenal gland to initiate the stress response. And again, it's also true in humans. The first thing I learned in medical school, everything has been published but not necessarily read. And there are papers in the literature showing that patients that have adrenal insufficiency have normal stress response. The problem, or not the problem, the fortunate finding is that in the absence of glucocorticoid, circulating osteocalcin is high because glucocorticoid is an inhibitor of osteocalcin expression. And obviously, when you stress these mice, osteocalcin goes higher. And so we ask if this increase is high, this hyperosteocalcemia of adrenalectomized mice explain why they can have a stress response. And for that, now we go back to the osteocalcin deficient mice, but we remove either one allele or two alleles. Obviously, two alleles, there is no osteocalcin. But one allele of osteocalcin left, circulating glucocorticoid are normal. Circulating osteocalcin is normal in adrenalectomized osteocalcin heterozygote mice. And then we stress these mice, and they have a normal stress response. Whether we look at heat, energy expenditure, heart rate, respiration, if you normalize, if you remove the hyperosteocalcemia of adrenalectomized mice, they cannot have a stress response, which indicates that osteocalcin is not only necessary for the stress response, but it is sufficient to mount an acute stress response. And in summary, because I did not show all the data, stressors act upstairs in the basolateral amygdala. Then they use glutamate as a neuromediator that acts on osteoblasts to release osteocalcin, that inhibits the parasympathetic tone, relieves the brake on fight and flight, and then if a lion comes into this room, we will all leave. So I want to tell you one more slide, because I just don't want to tell you that there is only one way to look at evolution. There are other people that think that bone have another function, and I let you with this slide. That's it.
Obrigado.